Happy Friday. As the case may be, it'll seem like Friday. Tomorrow it'll seem like Friday, and then Friday it'll seem like, oh crap. Everything making sense? I know you guys all went home and you looked at that stuff very carefully, right? No, you didn't, did you? Tell the truth. This is the quietest BB350 class I have worked with. I don't know. I got to do something to light your fire, whatever that is. I don't know. Well, last time I talked about water. I talked about weak acids. I talked about the Henderson-Hasselbalch equation. And today I'm going to put all those together in the form of a word that you heard in the song that we sang at the end of the day. And that word was buffer. It's a word that we hear a lot about. And it's a word that's a very, very important word from the perspective of our cells and from the perspective of biochemistry. Okay? So, buffer. When I say buffer, okay, the most common thing that people think about is the aspirin known as bufferin. And the aspirin known as bufferin got its name because people used to believe that aspirin, which contains acetylsalicylic acid, you hear the acid part there. When they heard the acid part, they said, okay, well, people who take aspirin, that acid irritates their stomach because some people have a problem with taking aspirin and their stomachs. Well, it turns out, of course, that your stomach has a heck of a lot more acid than is contained in one tiny little aspirin tablet. Okay? But nonetheless, the idea of buffering, which is where bufferin got its name from, the idea of buffering was important because it meant that you could somehow control the strength of an acid. Okay? Well, as I said, that's baloney. But bufferin is still a very popular aspirin. And the phenomenon of buffering, which is what we'll talk about, is a very, very important thing. So I'm going to give you some perspectives on buffers today. And I'm going to go through a little bit more what I did last time in terms of thinking calculations. Okay? No, well, I just remembered I meant to check my camera, and I didn't get around to that. Sorry. If a camera's pointed the wrong way, then nobody's happy at me after that's done. So let's see. Oh, it's good. Okay. I'm pretty good about guessing where it's going to be. So, okay. So a buffer is basically a system of a weak acid and a salt. Okay? A buffer is basically a system that contains a weak acid and a salt. Okay? A system that contains a weak acid and a salt, okay, is able to resist changes in pH. A system that contains a weak acid and a salt, a system that is a buffer, is able to resist changes in pH. How might we change pH? Well, we could add protons. If we add HCl, we'll be adding protons because HCl completely comes apart and we get all those protons, right? We could change the pH if we added a lot of hydroxides, like if we added a strong base, like sodium hydroxide that I talked about the other day. Okay? That's two ways that we could change the pH of a solution. But what we're going to see is that if we add protons or we add hydroxides to a buffer, the pH changes very, very small amounts as long as we're within the range of our buffer. We'll talk about what that. The strength of a buffer. How strong is a buffer? How much capacity does a buffer have? That's really something that we have to weigh when we're thinking about an ability to resist a change in pH. Okay. Well, let's get back to the Henderson-Hasselbalch equation, and because that really is the root of what's going on inside of a buffer. Okay? Now I'm going to give you this equation on the exam. You don't need to memorize this equation. As I said, I will give you every equation that you need for the exam. There are no equations you'll need to memorize. All right? This one you're going to hear a lot about. If I turn it on. This one you're going to hear a lot about. Okay? because it really helps us to understand what's happening in that buffer. Okay? Going to understand what's going on in that buffer. Now, as I told you last time, it tells us that the pH is related to the pKa. What did I say the pKa was? What did it measure? Strength of a weak acid, right? So it's related to the pKa and the logarithm, the concentration of salt divided by the concentration of acid. Right? And I gave you some rules about logarithms that I hoped you learned, and that was that if the number of a logarithm was greater than 1, the logarithm was positive. The number that we're taking the log of is 0. The logarithm, is 1. The logarithm is 0. Got to get it right here. And if the number that is the log is being taken of is less than 1, the logarithm is negative. Okay? Those three things allowed us to do mental problems about this buffer the other day. Okay? Okay, that's the root. Now, we'll come back to that, but before I do that, I want to show you something. I want to show you something we call a titration curve. You probably had to make these in freshman chemistry. Am I correct? No? Yeah, okay. Yeah. Kind of a pain. You keep adding it. Nothing's happening, right? And then all of a sudden it goes boing, right? And your TA is going, what in the hell did you do, right? One of the common experiences in freshman chemistry. Okay? Well, what that experiment that you were doing in freshman chemistry was aimed at teaching you was something about how a buffer worked. So I'm going to tell you here. All right? Let's imagine that I've got a system of acetic acid and acetate. Okay? Acetate being the salt. There's the salt. And there's the acetic acid right there. And the difference between those two is exactly 1 proton. The difference between an acid and a salt is always 1 proton. Okay? The acetate, which is this guy up here, has lost the proton. The acetic acid, which is right here, has the proton. Notice I will call this an acetic acid buffer, or I may call it an acetate buffer, but when I use the buffer term, I'm referring to both at once. Okay? Whenever I say the word buffer, what I want you to have pop into your head is the following thing. Buffer equals acid plus salt. Buffer equals acid plus salt. Because you have to have both in order to have a buffer. Now, I'm going to show you why in a second you have to have both in order to have a buffer. Okay? Everybody with me? All right. So, let's imagine, if you will, that we look at this curve and we see, well, this curve kind of comes up from down here, meaning the pH is rising fairly rapidly. It flattens out, and then over here it takes off and rises fairly rapidly again. We're only seeing a part of that overall solution. Most of you are going to look at this graph and you're going to memorize that shape. Okay? And that's okay. That's okay, all right? But if you understand what this shape is telling you, you will really understand what a buffer is doing. Okay? What are we doing in a pH mixture, or when we're doing a titration? Well, when you did a titration in freshman chemistry, they gave you a pH meter. They gave you a solution that had either typically a very low pH or a very high pH. We're in this class going to start with a low pH. Okay? And then you started adding something to change that pH. In this case, we're going to add sodium hydroxide because we're already starting out at a very low pH. We've got plenty of acid there. Okay? If we look at this, what we see is that if we move to the right just a little bit, the tiniest amount of OH that we added changes the pH from very low to a big jump. Just a very tiny amount. How much did we add? We only added the amount going from right there to right there. That's a very tiny amount of OH being added. But the pH went from down here about 1 all the way up to here. Okay? We saw the pH change fairly drastically. It changed over 2 pH units even though we only added a tiny amount of sodium hydroxide. You would look at that and you would say, buffering really isn't going on there. And that's exactly correct. In this region of the graph, buffering is not going on. The pH is changing drastically for just a little bit of OH. Okay? And I said that a buffer was something that resisted change in pH. This thing isn't resisting change in pH very well at all. All right. We get up a little further, and we discover that the more and more OH we add, the smaller that jump is. If we add an equivalent amount to this, we go from here over to here, we discover that the pH goes up maybe a half a pH unit. And if we add it again, we discover maybe it goes up a quarter of a pH unit. And in fact, what we see is we keep adding more and more, the smaller effect, the smaller the change in pH actually is. We're getting into what I call the buffering region. The buffering region is the place in this curve where adding OH has very small effects on the pH. And that was the definition I gave you for a buffer. It resisted those changes in pH. Okay? Well, we discover way over here, we get over to the other side of this graph. We keep adding and adding and adding. In each case, we're adding more and more OH. We see we go through this sort of flattened region over here, and over here, what happens to the curve? It starts to take off. Do you suppose that we're getting out of a buffering region over here? The correct answer is yes, Kevin. Okay? Yes. We're getting out of the buffering region. So it tells us that for a given, congested today, for a given system of acid and salt, buffering will happen within a certain pH range. What's that range? Well, I'd like you to look at this right here. There's a point that's labeled right in the middle, and this point that's labeled right in the middle corresponds to a pH of 4.76. Does anybody remember why that number is significant? Have you heard that number before? It was the pKa of acetic acid, exactly. Okay? Now, we're going to learn a bit more about pKa here. Right? 4.76 was the pKa for acetic acid. You can see on this curve that the flattest part of the graph is where the pH is 4.76. Everybody see that? Adding more OH has the smallest effect on the pH right here. All right. Why is that the case? Well, let's go back to our Henderson-Hasselbalch equation. Henderson-Hasselbalch equation says that the pH of a solution is equal to the pKa plus the log of salt over acid. Right? And I just told you that the maximum buffering was going on when the pH was 4.76. Right? We all agreed the curve was flattest where the pH was 4.76. So if I plug this in and I say 4.76 equals 4.76 plus the log of salt over acid, what does that tell me about the log of salt over acid? It means that the ratio of salt to acid must be 1. You've just learned something about a buffer. A buffer is at maximum capacity to buffer when salt equals acid. Okay? Maximum buffering capacity happens when salt equals acid. Pretty cool. If I told you that I had formic acid, and I said, draw a titration curve for formic acid, and I told you that the pKa for formic acid was 3.75, you probably have a pretty good idea about how to do that, right? The shape of the curve would be the same. the curve would start down here, and usually, by the way, this book doesn't include this portion, but it's okay to include that portion on your graphs. It would go up, and it would start to flatten out, and it would have its maximum flatness at a pH of 3.75, which is the pKa of formic acid. Everybody understand? Okay. So you can memorize that shape, but if you understand what's going on with the buffer, you're in better shape. Now, we can look at this buffer, and I can say, okay, well, how about I'm right there, where I have my pointer? Do I have more salt, or do I have more acid? I have more salt. How did you determine that? What's that? The pH is going up, but how did you determine that? Okay, the answer is that you determined it from the Henderson-Hasselbalch equation, but you weren't thinking about it. The Henderson-Hasselbalch equation told you what? pH equals pKa plus log of salt over acid. If I said this point right here, is that pH above 4.76 or below 4.76? It's above, because when I read it off, it's about 5 something, right? And what does that mean about the log term? The log term has got to be positive, meaning there's got to be more salt than acid, right? Good, good. Okay, this is great. This is great. All right, so you can read these curves and understand something about what's happening in that solution. That's really cool. Okay, let's take a look. Questions before I'm going to just jump forward. So questions about anything that I've just said so far. You could draw a curve. You could tell me on that curve where you had more salt, where you had more acid, I hope. You could tell me where maximum buffering capacity was, right? And you related these things to the Henderson-Hasselbalch equation, and you didn't use a calculator. Okay, you guys are great. Let's take a look at a bunch of acids. I showed you this the other day. Okay? I showed you this guy the other day. All right? Here's acetic acid. There's the acid, the HA. There's the salt, the A minus, and there's the pKa. If I told you to go draw a titration curve for pyruvic acid, the first thing you would say was, well, what's the pKa? I'd say it's 2.5, and you'd say, okay, duh, I know that. It's simple, right? And it is. It's very, very simple. Okay? If you guys learn that, I'm very happy. Very, very happy. Okay? Now, because you know that, you can do additional things. You know, professors are always pushing you to do additional things, and I'm going to push you to do additional things. All right? Let's imagine we're talking about carbonic acid. Carbonic acid is different than acetic acid, and it's different than pyruvic acid, and it's not different just because it has a different pKa value, but it's different because it has 2 protons that can come off. 2 protons can come off. Okay? Each one of those protons has a specific pKa. Do you have an idea about what that curve might look like? It goes up. It flattens out where? Flattens out at the first one. It goes up, and it's going to flatten out again. You guys can do this. Okay? Amino acids that we're going to talk about have this property. Every amino acid has at least 2 pKa values. Every amino acid has at least 2 pKa values. Some of them have 3 pKa values. Okay? Phosphoric acid down here has 3 pKa values. But it's nothing more complicated than what you saw up here. You'd have a flattening at 2.14, you'd have a flattening at 7.2, and you'd have a flattening at 12.4. We're going to use a rule of thumb in this class. The rule of thumb is if the pH of a solution is within 1 unit of the pKa, the buffer is working. That is, The buffering region for a given buffer corresponds to plus or minus 1 pKa unit around, 1 pH unit around the pKa. So if I said, what is the buffering region for acetic acid, you would tell me it's between 3.76 and 5.76. 1 pH unit below to 1 pH unit above. With me? If you go back and look at the curve that I showed you, you'll see that approximately that's actually what it looks like. Within about 1 unit below, 3.76, to about 1 unit above, when I add hydroxide, it doesn't change the curve very much. But outside of that, boing, up here, boing. I sound like I'm an airplane manufacturing company in Seattle. Okay? Makes sense? Okay. So pretty cool. Pretty cool. All right. We're also going to make, actually, I'll save this for later. Okay, I'll save this for later. But you understand the basics of what's going on in this curve, right? Very pleased with that. I see a lot of smiling faces. That's good. Okay. Let us proceed. All right. So pH turns out to be really important. pH, of course, is a measure of the acidity. pH is a measure of the proton concentration. And protons going on and off of things change the charge. They change the charge by 1 unit. If I go back to my acids, I look at acetic acid. The HA has no charge on it. The salt has a -1 charge. It has lost a proton. It has lost a charge of 1. 0 minus 1 is equal to -1. Every one of these guys that's on the screen, the salt has a charge 1 lower than the acid because the salt has lost 1 proton compared to the acid. When I look at things that have multiple protons that they can lose, what's the salt and what's the acid? The answer is it depends on the system that we're talking about. If I said that we were at a pH of 2.8 for malonic acid, what would be the salt and what would be the acid? 2.8, you would look over here for malonic acid and you would say 2.8 is about that one right there. It's within 1 pH unit of that one right there. So I must be talking about these two, and this would be the acid and this would be the salt. And if I asked you the same question, I said we've got a solution of malonic acid and we're at a pH of 5.5, and I said what's the salt and what's the acid, you would say there is the acid and there is the salt. And you'll notice that this is the same as this. Right? So something can be a salt in one system and be an acid in another system. That's not too complicated. Okay? We got 3 of them. All right? Again, it's going to depend. But the pH and the pKa will define which pair I'm talking about. Okay? All right. Okay. Now, pH is important. It's very important. Because I've already told you, I gave you an example on the first day. Imagine I have a protein that has a big hunk of positive charge over here and a big hunk of negative charge over here, and they're attracted to each other, and if they're attracted to each other, they're going to hang out closer together, right? Imagine I've just changed the pH where this one over here that was negative, attracted to the positive, moves from being negative to being 0. There's not going to be this attractive force between them anymore. The system is going to relax. And when it relaxes, the shape is going to change. And shape changes are everything with respect to activity of proteins. We'll talk about activity of proteins next week. But proteins have jobs that they do, work that they do, reactions that they catalyze. All of those things require a specific shape. And if I go messing with the shape, I ruin the activity of the protein. Okay? These two graphs I have on the screen are there to illustrate and tell you and show you that. It shows two very different enzymes. One enzyme on the right is found in your digestive system, further down in your digestive system. And you can see that we're plotting activity, that is, its ability to catalyze a reaction. What both of these enzymes do is they break down other proteins. The more active it is, the more proteins they'll break down. So if we measure how much protein is being broken down at any given pH for this particular enzyme called trypsin, we discover that at very low pH, it has very, very little activity. But as the pH is around 6 to 7, which is about physiological pH, we discover it's got maximum activity. Well, in our intestinal system, that's about what the pH is. And that's good. If we were to take trypsin and we were to put it into our stomach, what we would discover is we would have very little activity of trypsin because the pH of the stomach would be about 1 to 2 and we'd be way down here. When we're working with enzymes, we have to be concerned about that shape, and one of the factors affecting the shape is the charge. The charge is a function of the pH. Okay, everybody with me? All right. Here's another enzyme, pepsin. Pepsin is not found in your intestines. Pepsin is found in your stomach. And in your stomach, the pH is about 2.0. And when we look at the maximum activity of pepsin, it's around 2.0. If we put pepsin into an environment where the pH is about 7, we see we have very little activity. The pH at which pepsin works, okay, is an important consideration. So if I'm doing an experiment in the laboratory and I want to measure doing something with pepsin and I want to know how much protein pepsin can break down, if I don't do the experiment at a pH of 2, I'm not going to be using a pepsin that's very active. I'm not going to learn very much. Okay? We're going to see that there's a whole bunch of things that can affect protein shape. pH is the first of those that you're learning. Okay? pH is the first of those that you're learning because the more protons we put into a system, the more salt or acid we make. You tell me. The more protons we put into a system, the more salt or acid we make. The more acid. Now, I'm going to go back and show you something that I talked about the other day. Okay? It's right here. Here's what's going on. If I've got a system that has some acid in it, and it has some salt in it, and I start adding protons to it, what's Le Chatelier's principle going to tell us is going to happen? Something is going to gobble up those protons to reduce the concentration of protons. That is going to drive the reaction to the left. I'll have more acid, and I'll have less salt. I see some frowns. What the hell is he talking about now? This would be a good point to ask me some questions. Nobody wants to ask me any questions. I can just stand up here and just be lonely. I will. Okay. So, will I repeat what I just said is the question? And so what I just said was this. All right? This equation shows us the ionization of an acid. Acid loses proton, becomes salt. I talked about Le Chatelier's principle the other day, and I talked about it relative to sodium hydroxide, right? And what was sodium hydroxide doing? What was the hydroxide part of sodium hydroxide doing? Gobbling up protons. You get an A for the day, okay? Gobbling up protons. And when I took away protons, what did Le Chatelier's principle tell us happened? Something replaced the protons, and the thing that replaced the protons was this guy here, right? Now I've told you the opposite thing. Instead of taking protons away, I'm putting protons in. And when I put protons in, Le Chatelier's principle tells us the system is going to adjust for those extra protons. It's going to gobble them up, and the way it gobbles them up is with salt. With me? Okay. Now, this principle that I just described to you is very critical for understanding buffer problems. If you haven't looked at the buffer problems, and there's some problems that I have on the schedule page, I strongly recommend that you look at them. Okay? I'm going to tell you something about them here, but they probably won't make sense to you in some cases, and that's okay. It's okay to come and ask me. As I said, I don't bite. I'm happy to help you and happy to work with you. Okay? I said the other day that if I add for every hydroxide I added, I lost one proton, right? And when I lost one proton, this acid replaced that proton, right? So if I had, let's say, 50,000 of these that I started with, and let's say I started with 50,000 of these, and I added 10,000 sodium hydroxides. If I add 10,000 sodium hydroxides, I'm going to lose 10,000 protons, right? Yes, because the hydroxide will react with the proton to make water. And the system is going to say, whoa, better replace those protons. What's going to happen? 10,000 of these guys are going to give up protons. I'm going to have, at the end of that, 50,000 minus 10,000 of the acid, and 50,000 plus 10,000 of the salt. Because when this guy loses a proton, it becomes a salt. Conversely, if I started with 50,000 of, let's say I've got 60,000 of these and I've got 40,000 of these, and I put in 20,000 HCl's, that's going to be 20,000 protons. What's going to happen? 20,000 of these are going to gobble up 20,000 of these and make 20,000 more of these. I'll have 60,000 minus 20,000. Over here, I'll have 60,000 plus 20,000. Make sense? The rubber's hitting the road, guys. If you understand that, you understand everything you'll need to know about buffers. Now, the way you can tell, and by the way, this is going to happen a lot, okay? I find I talk to students a lot, and I really want students to learn things. But the most common reaction I get with students is they'll come to me and they'll say, I know that. I know that. And they'll get to the exam and they'll go, the exam isn't valid because I knew that. I knew that. I knew how to do that. Okay? And what they really mean is, you made sense. I understood what you said, but I didn't do it. And when they go to do it, they discover they don't know it, but they're convinced that they know it because what I said made sense to them. Don't take my making sense as learning something. Work the problems. Okay? Make sure you can work the problems. Make sure you understand the problems and what's in the problems. Because if you can do that, then I think you can reasonably say, I know that. But knowing that doesn't mean that when I say it, it makes sense. I'm paid to make sense, and hopefully I'm earning what I get paid. Okay? But don't confuse my making sense with your learning. Okay? Internalize it. It's very important for you to internalize it. Okay. Questions, comments? We're almost done with buffers, guys. All right. Blah, blah. Same thing. Okay. There's something you can memorize if you want to, but you don't really need to memorize it because it doesn't really matter. It's basically telling what you already learned, and that is if you had more salt than acid, pH is higher. If you have less salt than acid, pH is lower than the pKa, right? And if you have equal amounts, the pKa is equal to the pH, right? This tells you the magnitude. If the pH is off by 1, it's a 1 to 10 ratio. If it's off by 2, it's a 1 to 100. And if it's off by 3, it's a 1 to 1,000. If it's plus 1, it's 10 to 1. It's 100 to 1. It's 1,000 to 1. But since I'm not going to make you do calculations on the exam, you don't care about this, right? They're just numbers. What matters are the concepts. What you guys have learned are the concepts. Okay. All right. That's the last I want to say about buffers and pH at this point, unless you have questions or comments. I'd like to move on. We'll actually get a little bit ahead, which doesn't happen too often in the class. I'm going to talk about amino acids. Okay. All right. Let's talk about amino acids. All right. Amino acids. All right. Amino acids, as I said the other day, are the building blocks of proteins. There are 20 amino acids that are used to make essentially every protein on the face of the earth. There's only a handful of minor exceptions to that. 20 amino acids make up all the proteins on the face of the earth. Okay? You'll notice I call this the 3D world of amino acids because we really have to be thinking in 3 dimensions as we're talking about all of these different structures that are happening because remember I said shape is important, and shape doesn't exist in 2 dimensions. Shape exists in 3 dimensions. Okay? No, I'm not going to ask you to draw things in 3 dimensions. Let's think something about amino acids. One of the things you learned in organic chemistry was that if you have a compound that has a carbon in it, that has 4 different things attached to that carbon, that there's 2 different ways in 3-dimensional space that those 4 things can be arranged. They're called stereoisomers. Okay? Stereoisomers. Chemistry has a pretty elaborate scheme for naming things involving R's and S's and all kinds of stuff. Biochemists are, by the way, very lazy people. Okay? I'm a biochemist. I can say that. I'm a very lazy person. We don't use elaborate systems in there. In fact, you're going to like the systems that we use. We're pretty simple in the systems that we use. We call them D's and L's, which chemistry does as well. But you'll see we have a much simpler way of dealing with it when we have multiple stereoisomeric forms. Here's a compound that's got 4 different things attached to one of the carbons. One form is listed as D and one form is listed as L. Okay? So what? Well, it turns out that every amino acid except for one, and a web page that doesn't show up, every amino acid except for one has at least one carbon with 4 different things attached to it. Meaning that every amino acid can exist in the D form or the L form except for one. The one that doesn't exist in that form is glycine, and that's because, as we will see, glycine is a very simple amino acid, and it has no carbon that has 4 different things attached to it. So glycine can only exist in one form. Now, there's something interesting. Why do I care about the stereoisomeric form of this? Well, there's something really cool and interesting about the D and L forms of amino acids we find in proteins. Okay? If I were to take proteins from any one of you, if I were to take every protein from any one of you, or I were to take every protein from your dog, your cat, your chrysanthemum, or the bacterium floating or sitting on your skin eating away at you, if I took every single one of those proteins, what I would find is all of the amino acids would be in the L configuration. Every one of those amino acids would be in the L configuration. There's a minor exception with bacteria that I'll talk about later, but for our purposes right now, every amino acid found in proteins made inside of cells is in the L configuration. What's the chances of that, right? What's the chances of that? Well, the chances of that, there's no chance involved. They're made that way. They're selected that way. If you were to consume amino acids in the D form, your cells would not be able to use them to make proteins. Because they grab things with specific structures, and they put them together in specific three-dimensional ways. D amino acids don't fit in the three-dimensional scheme that proteins are made in. Okay? Now, this turns out to be kind of cool. One of the cool things is, I told you guys about the spacecraft that's floating around the comet, right? Spacecraft floating around a comet right now. They recently detected nitrogen coming off of this comet. They put a lander on this comet. And one of the questions they asked the lander when it got to the comet was, what's the structure of the amino acids that you find on the comet? Do you know why they asked that question? I'll tell you. The what? If what?
If there are cells like living.
If there's evidence of biology, right? Because if we randomly chemically make amino acids, exactly half of them will be in the D form, and exactly half will be in the L form. That's if we're chemically making them. We go and make a test tube, we put them together, that's what we get. But if we give them to a cell, the cell is only going to use one or the other. In the case of Earth-born cells, they're only going to use the L. Martian ones, who knows, maybe D. But if we saw a bias, we would say, oh, wow, that's cool. Whenever a meteorite lands on Earth, one of the first things that people do, if they can get it before people handle it, is they extract amino acids from it and they ask the question, is there a bias for D and L? Because if so, it suggests that these were not made randomly, but actually there was a cell that was involved in that process. Kind of a cool thing, right? So we have one way of analyzing for evidence of extraterrestrial life, the bias of structure of amino acids. All right, let's talk about amino acids. I told you that you're not going to need to memorize the structures of 20 amino acids. There are some things about the structures you're going to need to know, and I will make that clear to you. All right? There are some things about amino acids that you should know. You should know the names of all 20. You should know the names of all 20. If I say methionine, you should know methionine is an amino acid. If I say mumbo-jumbo barb, I will hope you know that's not an amino acid. Okay? So know the names. I'm going to just step, and you'll see if you look on the screen that I've grouped amino acids into 1, 2, 3, 4 different groups. I think you should know the group any amino acid is in. Okay? Nonpolar, polar, acidic, and there's that word basic. I didn't say base, I said basic. Okay? Let's take a look at just a couple of them. All right? Here's leucine. Leucine is an example of an amino acid that is fairly nonpolar. And by nonpolar, it means that other than its carboxyl group and its amine group, and by the way, every amino acid has a carboxyl group, at least one, and every amino acid has at least one amino group. An amino group is an NH2, NH3 sort of thing. Okay? Every amino acid has one of each of those. Some have 2 carboxyls. Some have 2 amines. Okay? So with the exception, in fact, we can see there's the amine of this amino acid right here. There's the carboxyl. Okay? And there's the rest of it. I'm going to give you some names here of this. This carboxyl that you see is called the alpha-carboxyl. Every amino acid has an alpha-carboxyl. This amine that you see here is known as the alpha-amine. Every amino acid has an alpha-amine. Excuse me. This carbon that both of those are attached to is known as the alpha carbon. And every amino acid has an alpha carbon. So it's got an alpha carbon, an alpha carboxyl, and an alpha amine. And they are specifically carbon, carboxyl, amine. Every amino acid has a hydrogen attached to the alpha carbon. And every amino acid has an R group attached to the alpha carbon. Let me get you a better figure. Okay? Where did I get that? Oh. I think that's the one I want right there. All right. So I'll fix that and get it for you. Okay? You've got it right there on your thing, don't you? Yeah. Okay. The figures that I put out have it, but for some reason that link is broken. Okay. That means excuse me, that every amino acid has those four things I just described, right? All four of those things: alpha carboxyl, alpha amine, alpha carbon, and an R group. Well, since every amino acid has an alpha carboxyl, an alpha amine, and an alpha carbon, the ways that amino acids differ from each other has to be in the R groups. And that's what we're going to be talking about next. Okay? Everybody with me? I think it might be a good stopping point. I do have a song for you. This one I can't sing worth a crap, so I've actually got somebody to help me to do it. And you can sing along, though, okay? It's called There we go. Okay. All right. So somebody's going to help to sing this, and this is the amino alphabet. You might know the song. We'll talk about all that stuff next time.